Over 60 million individuals have been infected with HIV, the virus that causes AIDS. HIV/AIDS is a global health crisis, and although intensive research efforts have dramatically lengthened the lives of infected individuals, a true vaccine remains elusive. A major focus of HIV vaccine research is the envelope glycoprotein, which HIV uses to enter target cells. In this video, we demonstrate how we obtain structures of this important protein using an imaging technique called Cryo-Electron tomography. Beginning with purified HIV, we show steps in preparation of frozen specimens, steps in recording electron microscopic images from these frozen specimens, and steps in converting these images into structural models that could be useful for HIV/AIDS vaccine design. A unique aspect of what we present in this video is that the work will be demonstrated by members of our laboratory spanning a wide range of ages, from middle and high school students to college and graduate level students, all the way to postdoctoral fellows and senior scientists.
The preparation of vitrified virus samples suitable for cryo-electron tomography is accomplished by spreading an aqueous suspension of viruses across small holes in a carbon film supported by a copper grid, and then rapidly freezing the viruses. The goal of this step is to prepare a frozen specimen of intact viruses captured in a near-native state. To begin, the grids are placed inside a glow discharge unit. This device cleans the grids by exposing them to ionized gas. Now that the grids are ready for use, a suspension of viruses mixed with small gold particles is prepared. These particles